today I'm just going to share with you my views on using systems biology approach for therapeutic development. And I must say that several of the speakers today do use different aspects of the systems biology approach to their therapeutic development. So basically the goals, as we mostly all know, is that if you want to develop a therapy, it's to stop disease or reverse its effects and avoid toxicity. So avoid having the cure being worse than the disease. And oftentimes in oncology, this is a very fine line of where you're not sure if the cure is not worse than disease. And beyond that, we also have run into these, different side effects that come along with therapies, and not to mention that the cost of therapeutic development is going up, which is being passed on to the customer, especially here in the US. I know it's different in other countries, but the United States does drive the pharmaceutical industry because of the cost benefit analysis of developing drugs here in the US. You make more money. Yeah, and so just to kind of drive this point home a little further, I mean, these are the people that are most affected by these higher drug costs, and you can see that often the older you are, and or the sicker you are, of course, it's going to cost you more money, and unfortunately, tends to be that people that have less money tend to actually end up being sicker people, usually because their ailments keep them from, maybe having the kinds of jobs that they would like, that they could actually produce income that would allow them to be able to afford these things. Another problem in general, in the way pharmaceuticals have been developed in the past and even the recent days now, is that the overall return on development is going down, and this is over just a 10-year period. You see that in 2010 the basic return was about 10%. Now it's down to 1.8, and this trend is continuing to the point that we're running to a problem in the pharmaceutical industry where it's not going to actually be profitable to make a drug. And on top of that, it's becoming more and more expensive to develop a drug, which is obviously what's cutting into the yields. So, people tend to go way far back to the 50s. I tend to not want to go back that far in the past. I'll just focus on 2010, but, it costs, $1 billion used to be kind of the thing that people throw around for the cost of to develop a drug. And nowadays it's approaching more like, $3 billion. And so if you have these, great idea and you want to get it spun up, you really have to have strong ties with the investment community to even get anywhere off the ground, let alone get your product developed all the way through the commercial space. This is true for, all the, many of the major players in the pharmaceutical industry. Some are better at this than others in terms of, their efficiency. But in general, you can see from this graph, or showing here, is, the amount of money spent per sales. So a percent of sales on how many drugs you get developed. Some companies, again, are better than others. For example, Novartis, pretty good job. Only 16% of their total, revenues ended up getting them to produce about 13 in NMEs. So, it's basically a problem in the amount of money it costs to develop a drug these days has to go down in order for this to be a sustainable industry. So, kind of how did we get here? And again, many of the talks that I've seen today are actually addressing this problem directly, but, I must say, most of the companies, we're not the huge pharmaceutical companies, which, is both good and bad. It's good for us and bad that we don't have maybe as much money. But how do we get here? Well, a lot of people point out, well, maybe all the easy drugs have already been developed, or all the easy diseases, in quotes, have been, solved or potentially dealt with, which is, which we know is not really true. High cost of drugs, I think, in the early days of, developing drugs, there wasn't genomics, so you couldn't just punch in a gene and say, hey, should I try to target that thing? Oh yeah, let's go make a drug for that, with no real kind of phenotypic knowledge of what that gene might actually be doing. Oftentimes we use next-gen sequencing. We just look at expression levels or something like that, and then make assumptions about that being a good target. And unfortunately, these types of models are what have led to driving up costs, and also to really getting to the end of a clinical trial and having the FDA kind of look you people in the eye and say, this really doesn't do anything. Why are we going to approve this? And then another issue is just the basic structure of these large biopharmaceutical corporations. And I know they have gone through restructuring over the past number of years. But, you get, high end executives that call the shots of where the company's direction go, yet they may not actually understand any of the science and the innovation that's happening at the basic level. And another thing that, Anthony's talk earlier today, the reproducibility out there in the literature has also created major problems. Again, you get this concept of, you have maybe an idea about a target. This is a pure target-driven approach, and you unfortunately don't actually end up being able to reproduce what you originally designed your experiments around to develop a therapy for. All right, so how do you solve this? How do you deal with this basic drug discovery pathway of or drug development pathway, commercialization pathway? You have obviously drug discovery and development and preclinical phase. That's basically what the summits are centered around. And then you have your clinical research, FDA drug review. Everybody wants to get to there, and then of course get onto commercialization and post-market safety monitoring. So have this basic process, and again, a lot of these talks today have actually are addressing this area of you want to discover a molecule. So how do you do this? There's different ways. There's different omics approaches and other ways to go about it. You're looking for you figure out your dosing. What might be a mechanism of action. Dealing with side effects, taking it through your ADMEs. To try to kind of get to the next level before you start doing in vivo toxicology assays. So, then you step into, the preclinical research where, you're deciding to do in vivo or in vitro, toxicity studies, and also your drug metabolism, your pharmacokinetics. And so all these areas can be, improved if you take a more complete approach to the process, opposed to trying to, leap forward off of, finding maybe one gene target that looked nice and without a lot of like phenotypic kind of information to guide you through the entire way. So, what basically is systems biology, and this is kind of a funny thing because I was graduated from the University of Florida and started my postdoc at Harvard Medical School in systems biology, and that was at the medical school. There's a completely different systems biology at Harvard main campus. And if you ask somebody from main campus what systems is, they'll tell you one thing. You ask somebody from Harvard Medical School what systems is, and they'll tell you something else. And that's probably true if you go to Caltech or MIT or something like that. So it's kind of a interesting kind of bag. But the NIH definition here for you is, basically what you're trying to do is create a total picture of what's going on, whether that's the complete organism level, the tissue level, or the cell level. And instead of taking the reductionist approach where you're just trying to, find out what a broken thing does and put it back together, instead you're trying to take all the parts that you understand, piece them together, and build some sort of model, mathematical model. And usually this is done in computationally powerful systems. With the advancement of AI, and really, it's not even advancement AI, it's really the advancement of, computer power, and the ability to have, a computer in our hands these days is more powerful than any computer on Earth, only a few years ago. So that leads us to kind of this ability to put together bioinformatics, all your omics approaches, cell biology, computational biology, and AI, machine learning, and that effectively will get you to a systems biology approach. So, again, the areas where it can be most effective are, in the two areas that we're talking about here today, which are in the discovery and development process and the preclinical, research stages. So, it's kind of, this is just taken from the KEGG database, looking at the, what they say is the pathways in cancer. And so, obviously this is very complex, but the general idea is to try to, build models around each of these, nodes, if you will, these different proteins and different areas, and to better build out where, we can see or develop a model, I should say, to actually then make predictions about if we would either maybe enhance or stop a particular protein from working or interacting with another protein, and how that would then be able to create a kind of virtual phenotypic assay, if you will. And then to test those kind of in silico assumptions in the real world. And this is obviously taking off in biopharma as, rolling AI into this concept of systems biology. More and more pharma companies are investing in AI in their drug discovery. And obviously we've seen again today several AI-based companies or computationally focused companies. And then similarly, the venture capital world is also, getting, more involved with putting money in AI startups that are, obviously COVID's kind of tamped down some of the deals, but I'm sure, once people are more comfortable about interacting and getting deals done, and really just the kind of risk map is a little more stabilized in terms of for investment dollars, then we'll definitely be able to probably see this continue to tick back up. So just in general, this is from a paper a few years ago, and drug discovery today, just outlines some of the methods that can be used and implemented in systems biology. Really it has to do with, again, building these computational kind of roadmaps in these models that you can use to actually make predictions, prior to maybe even stepping into the lab. And so you can already start to pre-screen ideas and test them before really you put a lot of money into the wet bench work, which is actually what is what really drives up costs immediately. And so just kind of quick overview here. Looking at some ordinary differential equation and partial differential equations, you start to build these kind of mathematical expressions that speak to proteins interacting or enzymes functioning or what have you. And so, right. So kind of again going back to the historic kind of way things were done. Initially, we didn't have sequencing, so you actually had to ask the question of, what am I biologically observing, and if I treat that observation, do I get a response? Typical kind of phenotypic assays. More recently, and since 2000, so around between 2000 and 2010, I think we kind of fell off the rails in terms of everybody started just going, oh, we'll just use molecular genetics. We got a target, bam, let's go, high throughput sequence, bam. The only problem is this has led to driving up costs, and then when you get to FDA, there's no approval, or you get higher failures in the clinical trials. So the idea is to step back, kind of bring modern kind of computational power, and with more modern throughput, yet also starting kind of back, taking a step back to maybe earlier days of drug development where you actually had to have more of a phenotypic kind of readout to really get to where you wanted to go, and even as a starting point, and as a way to actually use those assays to start to find compounds, opposed to saying, well, we're just going to make this compound hit this target and that will work, without a lot of data to back those ideas up. So the general basis is, and this is again from a few years ago, Nature Reviews Drug Discovery. The basic idea is you lower the costs in general, and you also increase the amount of speed at which you get to your end goal of commercialization. So just in general, and this kind of wraps this back up, when you have this cell systems biology approach or systems approach, you kind of reduce straight off the top some of these initial kind of target ID and validation kind of steps, because you're doing them simultaneously already in the screening and the optimization points. Your preclinical is probably going to be effectively the same. Later on, you will have to ID the target, but that's after you already have a fully functionalized assay to get there. And as we've learned recently, those finding what targets a drug hits when you already have a good readout is much simpler. Clinical development is probably about the same, yet you're probably less likely to then fail in this phase of development than you would if you're using some of these other kind of genomics, purely gene-driven concepts. And so the basic concept is shown here. You have a cell system, you test it out with your different drugs, and you can use a program, for example, this biomap profiling, and kind of start to get hits and understand what's going on a little bit better with your system in order to improve development. So again, this just kind of compares things. You build a model. It takes time. If you put omics, also is a little slower, and you may not actually get a true commercialized product at the end of the day. Whereas if you kind of fold them together and you input omics kind of data with modeling data, into a cell systems biology kind of level where you create the full system of understanding, you get much better, faster, and more reliable results. This is just a diagram to illustrate kind of those points, and it's an iterative effect step where you can, test your model, test it, validate it, see if your model is predictive. Yes, then you're kind of good to go and ready to step to the next level. So this is kind of bears out in the data or in the literature as well. Your blockbuster drugs are your first in class, and those are generally found through phenotypic screening models traditionally, whereas your follow-on drugs, which are going to maybe get you commercialized, but probably you're going to be in a more highly competitive market space, and therefore you're going to be making less money, is more based on the target based. So at Cellanyx, we do kind of combine live cell imaging and artificial intelligence. This is just an example of some of our assays that we run. But what we couple is biophysical biomarkers with molecular biomarkers. And we're able to use those to make observations about cellular behavior. So this is the movie of cell spreading. They're spreading out on a well-defined extracellular matrix surface. This is a DIC image taken at 20x of primary prostate cancer cells. So we work directly on primary cells. So you can ask questions about cells that are directly from patients that have not undergone the various things that cell lines go through in the development of cell lines. And so you can actually in real time screen patients for particular drugs. And then you can do different imaging timescales. So you can ask questions about not just cell spreading, for example. You can look at higher time resolution, and you can start asking questions about membrane fluctuations and how the cells behave in depending on what their if they're cancer cells or non-cancer cells, or how bad the person's particular cancer is. They will behave differently on the surface, and they'll have different kind of phenotypic response that you can then interrogate and ask the question of, if you add particular drugs, will it be effective for those patients? And then you can kind of build a panel of patients and ask questions about their omics around your phenotypic study and kind of combine these things together. And then you really have a more complete picture of the entire system. Another example of watching cells over longer time scales, you can see the different mobilities on the surface, just as some other examples of some biophysical biomarkers. And then this is a paper we published a couple of years ago, just showing demonstrating the platform, how it works. In this particular case, we were actually using it more of as a diagnostic tool. And so we were able to show using prostate cancer samples that, we're able to measure all these various biophysical biomarkers, couple them with various molecular markers, staining, for example, focal adhesion kinase, and understand how the cells are interacting with the matrix, and how the different cells, either cancer or non-cancer, were affected by the interaction with the matrix. And so how we do this is basically we just image the cells again, have an imaging regime, and that can be modulated and changed depending on what type of cancer maybe we're looking at, or what type of disease we're looking at. It's not strictly just for oncology, but really any disease state. Again, the caveat is that we do need sample from patients. We do need some cells from the patient. Um, but that generally is a good thing, because then you can directly interrogate the patient sample, directly, opposed to having to make inferences about a cell line, how that's going to work with a particular patient or a particular population. And so those data are collected again through live cell imaging. That information is all then, all the biophysical biomarkers, molecular biomarkers, are measured using machine vision software that we've developed in-house. And those data are then fed to a machine learning algorithm that then correlates the biomarker data with patient post-surgical outcomes. And so what we're able to show, or we're able to tease out in panel B, just real quick, is the difference between positive and negative cells themselves. So we're looking at cellular level, single cell, and then to put that back to a full sample level or patient level and make predictions. So our predictions are pretty good in terms of the sensitivity and specificity, or in the 0.85 plus range for both prostate and breast cancer. We also have data in kidney, bladder, and lung as well. And so really, just by taking this kind of approach where you develop a true phenotypic screen, and you're able to actually then build out a system that allows you to make more accurate predictions, or very accurate predictions. And there's ways to actually improve this, to make them even more accurate, that we still are in development. So just some sources, various sources that can be used to do these various systems development. At the admin level, there's these various kind of predictive programs. Biomaps is great. All these various different databases are wonderful to mine. Of course, we just heard from Silicon Therapeutics, what they're doing is amazing. Also put Nanome in there as well, as another great tool. BC Platforms is another tool that can be used for real world data mining and potential drug repurposing. And this is just some more potential sources of software packages that can be used. This comes from the same drug discovery review I was referencing earlier. So thank you very much for your attention today, and for the speakers that went before me, and the rest of the group that's going to come after. I'm sure the panel discussion is going to be great. With that said, I can take any questions, and happy to interact with you offline or directly. My email is michael@cellanyx.com.
Wonderful. Thank you so much, Michael. And it's always a pleasure to have you present. And I know many of our audience members have already connected with you via email.
Yes.
Your company's doing great things. And I believe your CEO, Dr. Guy Fish, as well. He, I think, participated at our medical device and diagnostics summit back in August.
That's correct.
Thanks again. And like Michael mentioned, if you are interested in connecting with him, his email is up on the screen, or you